Hello everyone, my name is Libby and today I will be your local expert on performing Nanopore direct RNA sequencing. Direct RNA sequencing with Nanopore technology is a method for analyzing RNA molecules offered by Oxford Nanopore Technologies. In this method, RNA molecules are sequenced directly without the need for reverse transcription to convert RNA into complementary DNA. Nanopore sequencing involves passing individual RNA molecules through a tiny hole on a membrane called a nanopore. As the RNA molecule passes through the nanopore, changes in ionic current are measured, and these changes are influenced by RNA bases as they move through the pore. There is a variety of controls you can use for these experiments. Some examples are negative control, where there's no RNA sample added to the sequencing reaction, and that helps assess the level of background noise and potential contamination. You can also include a positive control, like a well-characterized RNA sample, to validate the entire workflow and assess the efficiency of your library preparation. Spike-ins can be used to monitor sequencing accuracy, assess quantification, and evaluate the detection of specific RNA species. Or you could perform replicate sequencing of the same RNA sample to assess the reproducibility of your results, which can help identify technical variability and confirm the consistency of your data. Here are a few studies that used these techniques. This first study's goal was to explore native nanopore RNA sequencing of a human poly-A transcriptosome. This team isolated and sequenced the native poly-A RNA, and the process is shown here in Figure 1. The total RNA was isolated from human B lymphocyte cells, followed by bead-based poly-A selection. The poly-A isolate was adapted for nanopore sequencing by first attaching a proprietary ONT adapter to the poly-A RNA using T4 ligase, then generating poly-A RNA DNA duplexes by reverse transcription, then followed by litigating the adapted poly-A RNA strand into a second proprietary ONT adapter with an RNA motor protein, and finally loading the adapted poly-A RNA onto individual flow cells for sequencing using standard ONT protocol. I did mention that reverse transcription is not required. According to Workman, the generation of the complementary DNA with reverse transcription was not required. They did it to improve the throughput. This next study describes direct nanopore sequencing of full-length ribosomal RNA from E. coli without reverse transcription at all. They were able to identify 7-methylgluconine and pseudouridine modifications in ribosomal RNA, shown in panel A. And in panel B, they depict the ionic current versus the time trace during translocation of an individual 16S ribosomal RNA strand from this E. coli library through a nanopore. Upon capture of the 3' end of the adapted 16S ribosomal RNA, the ionic current transitions from an open channel to a series of discrete segments characteristic of these adapters. This is followed by ionic current segments corresponding to base-by-base translocation of the 16S ribosomal RNA, and according to Smith, this trace is representative of thousands of reads collected for individual 16S ribosomal RNA strands from E. coli. This final example involves a report by Simori's team, who investigated gene expression of microbes for possible seasonal differences in a marine zooplankton community. According to these researchers, there is a need for a cost-effective monitoring program for zooplankton sampling and species identification. They discovered that the most abundant mRNA transcripts coded for essential metabolic processes and that the genes involved in translation-related processes were the most expressed. And though this was a small study, these researchers gave a solid basis for other types of biomonitoring programs. Direct RNA sequencing with nanopore technology is a powerful tool for studying RNA molecules, but it also has some limitations. Nanopore sequencing, while highly accurate, still has a higher error rate compared to some other sequencing methods, particularly for insertion and deletion errors. This can be a limitation when trying to accurately identify RNA modifications or rare sequence variants. It can also detect RNA modifications, but might not be able to distinguish between different types. The sample preparation can be complex and time-consuming, and analyzing nanopore sequencing data can be computationally intensive, and it requires specialized tools. Handling long-read data and correcting errors can be particularly resource-intensive, and some labs may require significant infrastructure to handle it. But despite these limitations, nanopore technology has made significant strides in addressing many of these issues, and ongoing research and development aim to improve this technology even further. And speaking of advantages, here are some advantages of using this method over other RNA sequencing methods. When considering whole transcriptome RNA sequencing, the advantage of direct RNA sequencing with Nanopore allows for full-length transcript sequencing without the need for reverse transcription, which preserves valuable information about RNA modifications and reduces bias introduced during cDNA synthesis. When using poly-A RNA sequencing, direct RNA sequencing does not require any poly-A enrichment, making it suitable for sequencing poly-A plus and poly-A minus RNA molecules, including non-polyadenylated RNAs and circular RNAs. And for capture sequencing or targeted RNA sequencing, direct RNA sequencing can be used for capturing specific RNA molecules or genomic regions without the need for cDNA amplification, which enhances the accuracy of variant detection and quantification. Direct RNA sequencing with nanopore technology is a powerful and innovative approach that complements and extends the capabilities of various next-generation sequencing like genome analysis, gene expression, profiling through RNA sequencing, and quantitative analysis of gene expression. Direct RNA sequencing with nanopore technology plays a significant role in advancing genome analysis, gene expression profiling, and quantitative analysis of gene expression. Its ability to provide full-length, high-resolution data at the transcript level is a valuable addition to the NGS toolkit. Researchers can use this technology to address questions related to genome structure, transcript diversity, gene regulation, and quantitative expression, providing deeper insights into the functional genomics of various biological systems. This approach is especially important when studying complex eukaryotic genomes and in applications such as understanding disease mechanisms or characterizing the transcriptome of non-model organisms. And that concludes my presentation for today. I would like to thank you all so much for your time. I know I used up an extra 2 minutes or so, but if you wouldn't mind leaving a comment with any questions or your thoughts on the presentation, I would really appreciate it. And thank you so much.